Hi, my name is Gregory Tompkins. Today is July 12th, 2019. We're going to be going over my microbiology week 2 lab. This is the bacterial enumeration, dilutions, and plate counts. I already went ahead and got our tubes ready. We have the tubes here and then my agar plates that I poured earlier today. They've been sitting a little bit. They're numbered. We have 1, 2, 3, 4, 5, and 6. Numbered just as well as long as the tubes. We have 1 through 6. I have already added 2.25 mLs of distilled water to each of the test tubes. We went ahead and got our yeast mixture here, and that one's, it's been sitting for over 10 minutes. There's a little bit of water in the yeast mixture. So what we're going to do now is we're going to go ahead and do the transfer of the yeast into the test tubes. We're going to do it 1 through 6. We're just going to kind of go over the process on video. So this is my alcohol mixture, or my alcohol, it's not a mixture, it's my alcohol. Labeled alcohol there. I'm just going to get this nice and sterilized. Alright. And then what we're going to do is we're going to get 0.25 mLs. I'm not sure if you can really see it on the camera, but it's labeled here, 0.25 mLs. We have the 1 ml, half ml, and there's 0.25 ml. So we're going to do the 0.25 mL here. So it's there. Then we're going to go into tube 1. Sterilize the pipette. Now what we're going to do is we're going to dilute it. So we're going to go back into that same one. We're going to take the same 0.25 and go into the second tube. Alright, so that was into tube number 2. And we're going to do the same thing. And into tube number 3. From 3 to 4. Sterilize the pipette again. And we're going to repeat this process through all the tubes. Oh, what am I doing? This is going to go from 4 to 5. The balancing game with these tubes as well. So that was 4 into 5. This is the last transfer. We're going to go 5 into 6. Okay, so now that we went ahead and transferred all the yeast and diluted it from one to the other, the next process what we're going to do is we're going to add it to our agar plates. That's going to be part 2. It's the inoculating the plates. So again, I have these labeled as well. So we have 1, 2, 3, we have 4, 5, and 6. So we're going to do the same process. Actually not. What we're going to do is we're going to add from 1 to 1, 2 to 2, 3 to 3, 4 to 4. What we're going to do is we're going to pipe it 0.125 mLs. So what that is it says it equals out to about 4 drops. So we're going to do 4 drops from the number 1 tube into the number 1 plate. Then we'll do 4 drops from the number 2 tube to the number 2 plate, and so on, so on. Okay, so we're going to go ahead and start. Go ahead and sterilize the pipette again. We're going to start with number 1. So it calls for about 4 drops. Get a better grip on this. Here we go. One, 2, 3, 4. Alright. We're going to move this so it kind of evenly gets dispersed over the agar. Now that it's all evenly dispersed all over the agar, we'll let it sit. So basically we just swirled the plate gently to spread the solution evenly over the surface of the agar. So we did that with number 1 and tube 1. So we're going to go ahead and move on to number 2 and tray number 2. Okay, 4 drops. And we will just gently swirl the plate. Allowing it to evenly spread over the agar. Number 2, we'll proceed to number 3. Another 4 drops. Now on to number 4. hard to drop, really. There we go. And again, just gently swirling it. Four. Next is number 5. And the last one, number 6. Pipe isn't like it's not working here. Let's have a hole in it. No problem, I'm just gonna grab my other pipe. That one's probably just used a little too much. Perfect. Okay, now we have our plates poured and all of our samples smeared on the agar. So this is the first part of the lab. Okay, so now we have our 6 plates poured and they are ready for incubation. So we have all of our lids on. What I'm going to do to store them, once this is ready, I'm going to flip these over, and what it does is it secures the cap and it just reduces any chance of any contaminants or anything like that. So what I'm going to do is I'm going to place the agar plates in our incubation location in my cabinet, with the lid side up for 12 hours to allow the liquid to be absorbed in the agar. After 12 hours, we will invert them and incubate for an additional 36 hours. So we're gonna have these in the cabinet for the next few days. All right, thank you.